this is my cartoon wrap-up. If you watched the first iBiology talk of mine, you saw this before. I think it's a nice summary, though, that there's real genetics on the left. I think to pick on my oncohistone, what I just talked about, those are real mutations, hence the red asterisk. Those are real mutations in the DNA, but unquestionably, and in this case, even in a gene that encodes a histone, but they're impacting, on the right side of the coin, the slide, the epigenetic machinery, and in this case, this specific case, the writer of an epigenetic methylmark, and these sort of interplay of different modifications of histones, different modifications of DNA, they are reversible, and that's the hope. We can maybe then reprogram and get around these mistakes that are causing some of these disorders. So if you're really fascinated about epigenetics and I want to read more on the topic, this just happens to be a new textbook on epigenetics. It was put out this past year by Cold Spring Harbor Press, and it helps me to sort of, I participated in this exercise with some wonderful people that are the editorial team. And I think from Thomas Sinuan on the left, Monica Lochner, Danny Reinberg, this was in Danny's office in New York, myself and Marie-Laure Caparas, all of us felt passionate about trying to bring this together in a textbook form so that this complication and all of this complexity could be slowly digested by those folks who want to read more about your genome from the standpoint of the epigenome. Thank you very much. And I have a contributor slide I'd like to certainly take credit to where if I talked about anything that was sort of experimental or laboratory related, I showed a few pictures, but I have to sort of take credit to all the wonderful students, postdocs, and techs that have been in my lab over the years. You might have gotten the sense that we like to collaborate. We do. For example, the physician scientists with whom have given us tissue samples from patients. iBiology, the project, shout out to Ron Bale for getting me involved with this, and others. Alaska Foundation for hosting me today and the filming, the crew here, my funding, and Rockefeller University, and thank you for your time and attention. Thank you very much. Hello, my name is David Atlas. I'm a professor and head of laboratory at the Rockefeller University in New York, where my lab studies chromatin biology and epigenetics, and I'm very pleased to participate in this wonderful iBiology series. I hope that I can enlighten you today in one or both lectures of mine. What is epigenetics? Why do we care? And what is it that perhaps might be true about epigenetics such that it's not all our DNA alone? Is there more to our heredity than just DNA? I think we all grew up with the notion that real genetics is deeply founded in our DNA molecule itself. I want to use this slide to date myself and say that at Rockefeller University in 1944, it wasn't even known that DNA was the macromolecule that is the source of our heredity. That was work that was carried out by these researchers, Avery, McLeod, and McCarty. And on the left side of the slide, you'll get the sort of textbook notion that genes, the DNA molecule, really is the molecule that Watson and Crick described the structure of, that really is the basis of our heredity. And you can see in the yellow box, there are very defined DNA elements that lie upstream of genes. We often know these are transcription factors, transcription regulators. They can be positive or negative, but they tell the genes then essentially they instruct the genes to either be expressed or to be silent. So the idea here is, could it be genes are everything? You know, a tour de force certainly in 2001 was the landmark sequencing of the entire human genome. It generated a lot of publicity, rightfully so, where now we had case-by-case information about our blueprint, our heredity. And there were many, many good reasons that the Human Genome Project was as exciting and got as much attention as it did. Genes might determine aging. And for me, someone as old as I am, that would be a really good thing to figure out how to improve that. Genes determine disease. Of course, that would affect many of us. And then genetic analyses might provide us a way to diagnose diseases and potentially develop therapeutic strategies, what we now know is personalized medicine or precision medicine. So these will be all good things that came from the Human Genome Project. So there are some questions that are sort of nagging that came from the Human Genome Project that I sort of like to address to set the stage for epigenetics. First of all, there was a surprisingly low number of genes in our sequenced human genome, only about 21,000. So that seems remarkably small. It may seem like a big number, but it's a small number, especially when you consider that it's about the same number of genes that are in frogs and fish and worms. And I think it's counterintuitive that we would be not including or needing more genes than those critters. Where would individual variation come from, especially if you had twins? If twins are genetically identical and they're not exactly alike in terms of their outward appearance or phenotype, where would that variation come from? Of course, we'd like to know when we traverse through our lifetime and we're influenced by outside factors, environment, diet, other things, does that influence the genome? It would be, I think, arguably, well, the genome's fixed, so that can't really explain variation that way. And ultimately, when genes really became scrutinized with precise human diseases, was it the case that in every situation you found a genetic lesion, a genetic alteration that accounted for the disease? And if the answer was, I didn't find find a mistake. I didn't find a change. How might that be explained by another property? So this brings us to the theme of what I want to talk about in my first lecture. That'll be the term epigenetics. It was coined by Conrad Waddington in 1942, and he's very famous for this contour map that depicted a ball rolling down a contour map hill. He was a developmental biologist who was interested in why in early embryogenesis can the same genome give rise to different cell types. And so as the ball rolled down either the left side of the contour map or the right side, it ultimately came to lie in different troughs. He envisioned that those would be different outward appearances or different phenotypes, but importantly, again, all from the same genetic material. And epigenetics stands for the Greek prefix epi, means in addition or above or something on top of. So something on top of or in addition to genetics. So I think here's a nice way to sort of simplify your thinking. If you take a gene and think about it in terms of an English word, then it would be that's genetics, that's genes. But as we know in English, you can stress how words are pronounced. Are they pronounced quietly? Are they pronounced loudly? Do you boldface or underline words to get added meaning? Well, you don't change the letters, so the words don't change, but how you read them changes by these simple changes, and those will be your epigenes. For those of you that are more tech-savvy, I'm not a tech-savvy guy, you might think of genes as being the computer hardware, where epigenes are the computer software. It's the same hardware, but what you're putting into the system changes the meaning. And from this cover of Chem Biochem in 2011, a very simple cartoon notion of a puzzle where the authors are trying, the artist is trying to show, I think, some of the exciting connections between epigenetics and very diverse parts of biology. So you can pick your favorite puzzle piece, but if I was picking a couple to be fair, developmental biology is where epigenetics sort of was founded, and then that's the upper right-hand corner, and the lower right-hand corner, these white pieces. What about diseases? Again, nothing would be more important than disease states that might not be explained by genetics, but might be explained by epigenetics. Another very useful way to think about a genetics situation, we're almost to Valentine's Day, how about a cake? We can all take a cake perhaps, and that'll have a certain taste, but if we literally put on different icings by our choice, it's the same cake, we haven't changed the cake bit, but just by adding different icings, sugar, non-sugar, you might be able to dramatically change the taste. And I think that's a nice way to think about epigenetics, and it just happens that frostings normally go on top of the cake. Think of the term epi on top of genetics. More appropriately in real biology, how do you take a young embryo with early cells being established that are all, again, pretty much undetermined, they all don't know quite who they're going to be or what they're going to become, and then the same genome, as it advances through development, is going to give rise to different cell types, blood, nerve, bone, skin, and now, once that has happened, these cells know who they are, and for the most part, they're determined, and they're going to give rise to daughter cells that carry on that identity. Blood cells will give rise to other blood cells and so on. As you know now, it's a very hot area of biology, there are small populations of cells we refer to as stem cells that are held in reserve, and they seem to have very much interesting potential because they're pluripotent. They can now still go into different cell fates. And a very hot area, of course, is can you take a fully differentiated cell and reprogram it and get it to go backwards into a more pluripotent state. So the big point I want to make is that all of these cell types, no matter what cell type they are, they all have the same genetic material. And sadly, I think very closely connected to stem cells, when you reprogram and you sort of make mistakes in this epigenome layer, you might actually be able to sort of push those cells backwards in development where they would take on a more undifferentiated property and take on more proliferative capacity. And there seems to be many, many examples now where those type of cells take on a cancerous phenotype that, of course, is often a very dreaded disease. So how do we really start to think about epigenetics now? Not so much with the cell biology, but how about the actual mechanisms? And this might make you think he's crazy. This is, I think, a wonderful cartoon that was drawn by one of my former lab members, Sean Taverna, who's now a faculty member at Johns Hopkins Medical School. And with apologies to the women who might be listening to this, the DNA molecule is clearly stepping out of the shower, and that DNA molecule is taking a choice. Should it grab a towel that's labeled histone, or should it grab a towel that's labeled herstone? And it looks like nature opted for the histone, so with apologies. So what do I really mean when I say histone? You go to a schematic drawing of the nucleus. Our DNA in higher cells is not naked DNA. It's in fact packaged by these proteins that kind of give a conversion of a bead on a string. These fundamental repeating units are termed nucleosomes. And what nucleosomes really are histone proteins that are acting as a scaffold around which roughly 2 turns of DNA wrap. And then you can see on the schematic cartoon, these beads on a string or nucleosomes will wrap further and further into higher order structures that truthfully are still being determined. But if you take a step back and you think about the structure maybe like a slinky toy, you might be able to understand right away that if you were to pull that slinky apart or find more what we term open or decondensed chromatin, that might be a better environment for genes to be expressed. Or in contrast, if you took that slinky toy and just compressed it, more like sort of all of that chromatin that's really packed together, that would be a harder environment for genes to be expressed. Even these transcription factors would have a harder time finding their DNA sequence to act on in that really compacted environment. And under some conditions of biology, and it's important for me to show you a real nucleus. This is a human cell nucleus stained in such a way that you can see dense and decondensed chromatin. This is an electron micrograph. And I hope you can see there are very patchy, very dense domains that are within that nucleus that are termed heterochromatin. That would be this compacted chromatin environment. where interspersed around these two regions are more open, decondensed chromatin is referred to as euchromatin. So here's a real micrograph of a real human cell. And now under some conditions, you can actually tease out that chromatin, float it, if you will, outside of the nucleus. And you can see now these chromatin fibers sort of in their nearly more visible state. On the lower panel, you can see the sort of classic bead on a string, like a pearl necklace, if you will. And somehow that chromatin, that more euchromatin, open chromatin, may be able to be flipped into a molecular switch, by a molecular switch, that'll compact that and turn that into the top panel, which is more condensed, compacted chromatin. So of course it became important for my type of lab, a chemistry lab, to try to ask, well, what would those molecular switches be? What would be the target of this sort of switch that could throw on and off states between chromatin, euchromatin, and heterochromatin? So this takes us to another Rockefeller, I think, very pioneering discovery. Here's a nice look at this individual I want to pay credit to, Vince Alfrey. And you can see that the nucleosome unit now in atomic structure, you can see in different colors the histone proteins wrapping DNA. I think you can just see some of the Watson and Crick helix wrapping on the outside of the DNA. But you do see these tail projections that are emanating outside of the DNA helix. And what Vince Alfrey, who's shown in the is shown a picture of, proposed in 1964 was that there would be chemical modifications, chemical marks that would be added to these tail domains that would then act as a switch. They would be a signal, if you will. They could instruct the cell to either be on or off. So it was his dream, his hypothesis, that these chemical modifications would potentially be that switch that I was referring to in the past slide. Now, of course, every chemical modification would have potentially the need for an enzyme system that would add that mark. And in my lab, we've nicknamed these writers, or there could be an opposing enzyme system that would strip these chemical groups off, which we've nicknamed erasers. And they would oppose. They would be the on-off, if you will, switches. And this was Alfrey's hypothesis, literally proposed over 50 years ago, 1964. Now, how would you access these enzymes? I think many of you would not have thought about this model organism. So on the top panel is a schematic cartoon of a ciliated protozoan that runs its life remarkably. It's very cool biology here with 2 nuclei. And a real picture of the tip behind that is shown below on a stain, so you can see sort of the organism on a slide. This is a unique property of ciliates that they run their life with 2 nuclei. The larger, and hence the name macronucleus, is the nucleus that's solely responsible for gene expression. This genome is packaged in a way that you might think of it as a massive euchromatin. And in contrast, the smaller, in this slide, micronucleus that's shown in red, it is very compacted. You might be able to tell that on the slide on the bottom. And it is completely transcriptionally silent. What's important about that is that nucleus does no transcription. It's reserved for an important feature in the organism that isn't really displayed here until it has the sexual life cycle. So you might think it's not even necessary, and that would be true here. It doesn't play a role in this case. Now, when I was a postdoc, I was intrigued by this system. I thought it was a little bit strange to work on something so odd, but I want to show you one gel line. So this is prehistoric. And what it is to the best of my ability, I turned trying to biochemically separate macro and micronuclei. And I joined a lab, Martin Gorofsky's lab, that was able to do that. They pioneered those methods. And what you can see here are 2 lanes of a gel. On the left, you can see the macronuclear histone protein displayed, and on the right, the micronuclear histone. And I hope you can tell, without going into the details of the gel system, there's a much more laddered sort of property or look to the macronuclear histone. And I've sort of tagged some of those ladders, the rungs of the ladder, with these green dots. And what was striking is you saw a lot more laddered effect, if you will, in the macronuclear histone preps that I made, and you didn't see nearly as much of that ladder in the micronuclear preps. That sort of box, that dash box, shows you where you don't see many of the laddered forms. So I thought this was pretty exciting, and I thought this might give me a handle on using this organism. What I need to now explain is that those ladders are due to charge differences, and each rung of the ladder is due to an increased chemical group, which is exactly what Alfrey proposed, which were these chemical groups called acetyl groups. That's too much detail maybe, but that's what I thought then would give me a good chance to go into the laboratory, purify the macronuclei, and isolate the writer that would be responsible for adding acetyl groups to the histones, as you can see here, primarily in macronuclei. So I started my lab group. I was able to attract a few great students into my lab, one of whom was Jim Burnell. And this was actually right from Jim Burnell's PhD thesis. Jim's long been out of my lab, but it was Jim's dream that he could sort of invent or use, I think, a quite ingenious in-gel activity assay that I'll quickly describe to you. Jim's idea was to put histones, the real proteins that I've been describing in my lecture, into a gel. So that's highlighted by the blue color. So he impregnated an entire gel with blue histones, not really blue, histones diagrammed in blue. And as a control, he put in another identical gel, non-histone proteins, that would be a control. And then the key was for them to take extracts from this macronuclei, from Tetrahymena, and run those out onto the gel. And in a perfect world, it was his dream that if he then sort of treated this gel with steps that I won't necessarily explain well, and then importantly, add the radioactive cofactor of the reaction, which some of us would know as radioactive acetylcoenzyme A. So the acetyl groups come from a metabolism, and he would do this, incubate this gel, wash it, and then in a perfect world, he might actually see where the enzyme